of a year. I'm slowly getting used to all this. Works well for teaching, not so well for your social life, I think. Well, thank you very much, guys, for inviting me. As Vlad said, I'm going to talk about nutrient and bioactive absorption and the role of intestinal mucus layer and perhaps some effects that dietary fibre of various sorts might have in terms of that interaction. So let's start by talking a little bit about the geography of absorption and digestion. Clearly, you can think about kind of some of the Pavlovian effects in terms of responses to food, but that kind of pushes slightly into the realms of psychology, and I'm not a psychologist. We have collaborations with the Department of Psychology for that, so I'm not going to talk about any of that. So we can think about really, we first start to come into contact with food through oral processing, and that's where we start to have release of aroma, and we start to sense taste and texture especially, and there's a little bit of digestion in terms of starch hydrolysis, but obviously mostly comminution and reduction of particle size through chewing. And although we never think about mucosal surfaces really in the mouth, they are nonetheless important, and that mucins have a role to play there, certainly in terms of lubrication and bolus formation, but that is a kind of topic on its own, and I could certainly give a talk on that, so I'm not really going to venture there. Subsequent to oral processing and formation of that bolus, food then passes down into the stomach for gastric processing that involves a certain amount of proteolysis and lipolysis, and obviously gradual acidification, but that's really a storage vessel. So it's good for triggering sensations such as fullness, and that may relate to appetite sensors, but really that's not the main center of digestion. So although there is a little bit of gastric processing in terms also of physical processing, so things like shearing and grinding, it's subsequent regions of the GI tract that are important for absorption. So it's the intestinal processing where the real proteolysis and lipolysis and amylolysis take place, and where mixing and transport and diffusion through the unstirred water layer, and then finally absorption, which is the critical bit that we need to deliver to in terms of nutrition. A part of that process of intestinal processing, there is also nutrient sensing, and the reason that's important is that it also links to hormone secretion that controls things like gastrointestinal motility, enzyme secretion, bile secretion, and appetite. So it feeds into the feedback loops that control rates of digestion in order to optimize our extraction of nutrients from the food that we're consuming. And because those sensors are, if you like, also part of the absorption system, anything that affects rates of absorption is going to affect the sensing systems and the feedback loops at the same time. So that's a kind of very broad brush view of the digestion steps that lead up towards absorption. So if we look in a bit more detail about what the absorptive surface looks like, this is an image of porcine intestine, fairly fresh from after sacrifice. And you can see, I hope, that it is very shiny and has lots of mucus on it. And that mucus is what is providing the protective layer of the underlying epithelium. And if we have a look a little more closely at that surface, we can see, let me just turn on my laser pointer. You can see that there, here we've labelled the mucus in green, and so there are lots of regions where there's mucus, and we're also looking into the crypts at the bottom between the villi. So we're looking between villi and into the crypts at the bottom, and you can see these circular structures with the mucus filling them. So throughout the spaces between the villi and down through the crypts, you see this protective mucus layer. And then if we look even more closely at the structure of that, we can see this network of linked chains that form a very coherent network. And I'll talk about that in a little more detail, because it's the way that that network behaves in different regions that is important for inferring its subsequent behavior. Of course, it's not a static system, so there is lots of stretch and contraction over the course of the meal cycle. So immediately post-meal, if you looked in the stomach, you'd see it was very stretched, and then after the meal has been digested and the stomach's been emptied, and the same is true to a large extent for the intestine as well, you see that it's shrunk. So the area that you're covering has changed, and you see significant rouging, so folding of the surface, and you also see that this thickness of this layer has changed because we're changing the surface. And you can imagine that as you change that through different cycles, you start to break up different types of structures. And if we look at what the thickness looks like and the structures look like, perhaps a bit more coarsely throughout the gastrointestinal tract, you can see that in the gastric compartment, so in the stomach, we see a relatively thick stratified layer with a loosely adherent layer on the top. And of course, the architecture in the stomach lends itself to that, because you have essentially flat areas with these gastric pits where mucus and other components are secreted. So this is the type of structure that remains relatively stable. Similarly, if I go to the other end of the GI tract into the large intestine, we see actually something that looks relatively similar. So again, you have relatively flat surface with these crypts that are essentially pits, and we have a stratified layer of mucus with a thick layer of loosely adherent mucus over the top. So actually the architecture of both the mucosal surface and the mucus is relatively similar in these two extremes, whereas in the small intestine we see very different architecture. So you see obviously the crypts, but you see villi as well, and the length of the villi changes as you go from the proximal to the distal small intestine. So as you go from close to the stomach down towards the large intestine, the villi get shorter, and this tightly adherent stratified layer is very, very thin or almost absent from a lot of this area. And why might that be? Well, it might be because when you have motion here, then there's less disruption of this stratified layer, whereas here, because you've got these long fingers of villi protruding into the mucus layer, they tend to break it up, and I'll show some evidence of that shortly. And also, if we think about the requirements of these different regions, they are rather different. So the stomach is not a site of absorption. A few things, ethanol, for example, get readily absorbed in the stomach, but not much else. Whereas the majority of our nutrients and other bioactives that are absorbed in the small intestine. So it's important that we have are mixing here, and we're able to transport these nutrients and bioactives to the mucosal surface for absorption relatively quickly. Of course, also in the gastric phase and the small intestinal phase, certainly in the more proximal regions, the bacterial count is relatively low. So it's not zero, but it's not at the high levels that you see in the large intestine. So part of the reason for this structure that you see here in the large intestine is to prevent bacteria from penetrating this stratified layer to start to become problematic to the mucosal surface. So you can have very high numbers of bacteria in the center of the lumen of the colon, and to a certain extent in this loosely adherent layer, but this dense stratified layer of mucus is essentially impenetrable under normal healthy conditions to most of the bacteria. At least that's the general consensus. So the reason that we see these different structures is because of the different architecture and the different requirements. So let's look at that stratified layer in a little more detail. So I'm not going to go into this in lots of detail because there's people on this call who are far more expert in this than I. But here's an example from the Johansson paper from 2013 that kind of outlines how this works. So you have your normal intestinal epithelium, so enterocytes, interspersed with goblet cells, and these goblet cells produce this stratified layer. And this is composed of these hexagonal structures formed from the MUC2 forming initially dimers, and then these trimeric links that come together in these hexagonal structures. And then in addition to this secreted mucus forming this dense layer, and by the way, I should perhaps mention that as this stratified layer starts to break down, it generates the more loosely adherent layer on the top. But this can also be relatively resistant to penetration and have a relatively high viscosity. And then linking this stratified region to the cell surface, you also have membrane-bound mucins that are tethered into the cellular membrane and kind of provide a continuum of this highly glycosylated glycoprotein region that provides the protection while allowing uptake of some small components. So in the large intestine, we're obviously talking about uptake of generally small components, whether that be free fatty acids, short chain fatty acids, or other small phenolic compounds. And we have shown evidence that a lot of these trimeric structures remain intact even in relatively purified mucin. So when we've removed this layer mostly from the loosely adherent, you can still see intact these structures by AFM. And they also tend to link up and form these networks again by AFM. So and lots of measurements have shown that typical pore size for these is around about 100 nanometers, maybe a little less. So that clearly limits any bacteria from being able to penetrate that because they're larger than that. But small molecules, well, I haven't said that, relatively large molecules, so tens of nanometers, can certainly penetrate through this area. And it's that ability to penetrate or not that is key to the functionality. So again, here we see an EM micrograph this time of this mucus layer, and you can see bacteria trapped in it, and this is draped over the surface of the intestine. So again, highlighting that the bacteria tend to get trapped in this network. So the question is, does that network trap other things in it apart from just the bacteria? and what happens when other components from food start to link into and combine with that mucus layer, because essentially you have a polymer system, and if you're increasing that concentration with a different polymer, does that affect the ability to penetrate? And we started thinking about some of these things a while ago, and started looking at the effect of a typical food polymer, sodium alginate. So there we had the question of whether the addition of sodium alginate into chyme in vitro decreased the diffusion of digester into the mucus layer. So if we make a digestion system with lipid in it typically, and we add sodium alginate to that as a stabilizer, one typically might have, can we see whether that makes any difference to the ability to diffuse through that mucus layer? And we saw that it did, but only under certain circumstances. So at the start of a digestion process, you have an emulsion where the droplets are typically relatively large, of the micron scale, certainly larger than this 100 nanometer pore size that I talked about. And I think you won't be surprised to know that those kind of large droplets can't penetrate through the mucus. But subsequently, after a couple of hours of digestion where you've got significant amount of lipolysis, then you can start to see diffusion coefficient going up. So here we see after 5 minutes and after 2 hours, and you can see diffusion coefficient significantly higher as the droplets are smaller, and you're looking at diffusion of more micellar material. And if we have look at this in the presence of alginate, you can see that you very much reduce the ability of the digester to diffuse through that mucus layer. So it looks initially like you have some effect on the permeability. And of course, we don't want to prevent the ability of lipid to diffuse through the mucus layer. That would be a bit of a disaster. Delivering lipid to the colon has side effects that I won't go into, but I can assure you can imagine what they might be. So we don't want to do that, but under a lot of circumstances, we might want to delay that kinetics of transport and absorption. We looked at this in a little more detail, and you do see an effect, but even at relatively low concentrations of alginate, that you don't see with in the absence of alginate. So it's quite clear that there are some effects to be had, and I guess that's not too surprising because we know that the alginate can diffuse into the mucus layer here. So here we're seeing the diffusion of the alginate into the mucus, and as we push up the concentration, that diffusion coefficient drops a bit, but it's still able to diffuse in. So even looking at this, it is clear there's some evidence that at higher concentrations, maybe you're clogging up the pores a little more and having an effect on the self-diffusion of the alginate. So there we're able to have more penetration into the mucus to have that effect rather than just forming a layer on the top. And I know that a lot of people have also started looking at mucoadhesives. So typically things like chitosans are used as mucoadhesive, but the issue with that is, of course, they bind just to the top of the mucus layer and are completely unable to penetrate, whereas these systems are more able to penetrate into the thickness of that layer in order to and block up the pores more generally throughout the mucus layer. So how do we put some quantification on these things? Well, one easy way is to look at rheological properties using the diffusion of probe particles. So typically we've done that and indeed lots of other people have done it using latex beads. Not a well-known food, but they do have the advantage that they're a very specific size. And you can measure the mean square displacement of these particles. So if we take an image we see the position of particles, and then if we take an image a little while later, you'll see that the particles have moved, and this enables you to track the motion of all of those particles. So you get a track that looks something like this, and from that we can measure the mean square displacement, and the mean square displacement is related to the diffusion coefficient, and of course you can then apply Stokes-Einstein to relate that to the local viscosity. Obviously, we're making a few assumptions, but this gives you a measure of the rheological properties at the scale of your probe. So at the in hundreds of nanometers, typically scale, or smaller if you're using smaller probe particles, which has the advantage over a rheometer where you're typically measuring it over millimeters at least. So it's much more relevant scale to what we're talking about. And again, this highlights what I was saying earlier about the effect of the alginate on a digester that's diffusing through. And the disadvantage of the digester, of course, is it allows us to give a rough measure of diffusion coefficient, but we can't get information about the local viscosity from that because we don't know the size of the objects that are diffusing. We just know that we're having an effect on the permeability. So it's not terribly quantitative in terms of its effect from that. So what factors might be affecting the ability of these different components to diffuse through? Well, as I mentioned, things like chitosan have been used previously as mucoadhesive because they carry positive charge, and mucin carries a negative charge. So you immediately have binding. But it turns out that one of the factors in certainly the small intestine that is important for that is the bile salts. So bile salts aid the digestion of lipid, and so they normally coat the surface of droplets, and they encourage the formation of mixed micelles, which are the much smaller objects that are able to diffuse through the mucus layer. But what about their effect on just the emulsion that's in the small intestine through lipid digestion itself? Well, in the absence of bile, if you just take a normal protein stabilized emulsion, you'll find that actually most of the droplets stick over the surface of the mucus layer and there's very little penetration. But when you add the bile, it absorbs to the surface of the droplets, significantly decreasing the charge, and you're able to see penetration at that point. So we can see that charge is important for governing the ability of components to diffuse through the mucus layer, and this is not too surprising. The other thing that helps diffusion through that layer, or at least transport through that layer, not diffusion through that layer, is mixing that's going on. So here we can see a small intestine that has been inverted so that the villi are on the outside so we can see them rather than on the inside, and the tips of the villi have been stained so that we can see them. And you can see that in the relaxed state, the black dots are quite a long way apart, as these villi have relatively large gaps between them. And in the contracted state, where the muscle is pulling the villi closer together, you can see the dots are very close together, and the villi are also close together. And the fact that this changes means that you get a flow in and out between the villi, and certainly in the small intestine, this is quite a significant mixing source. And if you don't believe that this actually happens, then let me see if I can show you. So this still has the muscularis in place and is showing peristalsis, albeit of the inverted intestine. And you can see there is significant movement and significant change in the villi spacing that adds to the mixing. And you can understand why you don't have a consistent striated mucus layer between those villi. because there's a lot of force pulling these apart. So when we look at the status of the mucus between the villi, it looks really heterogeneous. And that the reason it looks heterogeneous is because it's continuously being mixed because of this kind of process. So lots of motion going on, causing that mixing in the small intestine. And I think although you see contractions in the large intestine, the architecture is very different, which is why that striated layer is able to maintain its presence. There's been some modelling done, and here's a Limit al. Food and Function paper from 2015 looking at modelling some of this behaviour. Here they looked at the difference between a Newtonian and non-Newtonian fluid under these conditions where you see contraction and expansion, so of the gap between the villi, and you can see what happens to the fluid. And the fluid from close to the tips is drawn into the let me just turn my laser pointer on, is drawn into the gaps between the villi, and then as the contraction occurs, it's pushed back out again. So you see fluid being drawn in and out and mixing in the around the tips of the villi. And when we looked at this in more detail in ex vivo systems, so when we take tissue from a freshly killed animal and have a look at what's happening and what properties the different components have, you can look at to see whether you get penetration of these beads of different sizes into the interstitial gap. So can we see penetration into this gap between here, even though there's no peristalsis going on? And my colleague Adam Mackie used 500 nanometer latex beads and then looked at different heights, first of all above the tips of the villus and then gradually pushing in, and that's what you can see going through here. And they saw the ability of the bees to penetrate. So this mucus layer doesn't prevent even quite large objects. So 500 nanometer latex bees, that's kind of bacterium size, to penetrate at least, in this case, 100 microns below the surface and possibly further. Obviously, there are limitations to how deep you can go with high resolution microscopy in these systems. And here you see 100 microns in, and there are clearly the red beads being shown in the mucus between the villi, and here you see that in blue is the nuclei of the cells in the villi. So you can see that you get some penetration. Of course, one of the advantages of using the 500 nanometer latex beads is that you can do some quantification. So first of all, they looked at the proportion of particles that were able to diffuse. And on the surface, about half of the particles were able to diffuse and half of them were not, so they were trapped. So you see some trapping. And then as you go further down, you see more mobile proportion, but then this is perhaps not too surprising because the immobile proportion has already been trapped on the surface. So further down, you see a slightly higher proportion, so 40 microns, and then 80, you see higher proportions being, sorry, that should be nanometers, I'm sure, not microns, should be trapped. And then you can also measure the mean square displacement, and from that you can get a diffusion coefficient. And regardless of whether you were at the surface or whether you were further down between the villi, you could see the diffusion coefficient was the same. And from the diffusion coefficient, you could get out the viscosity, and typically it was 10 to 15 millipascal, so that's about 10 to 15 times the viscosity of water. So it's not just diffusing in water, but it's not a really viscous system. So it's not too surprising that you get the ability to diffuse down there. So what is it in these systems that is really providing the rheological properties? Is it just the mucus, or is it mucus plus other components? Now, one of the things that you may or may not be aware of is that the cells turn over relatively quickly, particularly in the small intestine. So they will travel from the bottom of the crypt to the tip of the villus here in something like 3 to 5 days. And when they reach the point here, they will be shed. So they will be removed from the tip and undergo apoptosis. So the cell dies, the hole is sealed up, but you are left with cellular debris around here. So in this image, you can see that the villus has been stained for mucus. So you can see the goblet cells very clearly stand out. And you can see the nuclei in red. And you can see the nuclei not just in the villus, there's lots of cellular debris out here that contains nuclear material. So we started to look at this, and we measured the diffusion coefficient of 500 nanometer latex beads at different concentrations in the mucus that we had removed from this region, and as you go up in concentration, at some point the beads are no longer able to diffuse, and this is a relatively high concentration of something like 200 milligrams per gram. If we then purify this, so we're only looking at the mucin component, we can see that we have a lower concentration required in order to bring those latex beads to a standstill. If we then just use the DNA component, you can see that even less is required. And this is kind of fairly sensible if you think about it. DNA is a really big molecule. And when it starts to degrade, it starts to unravel, and actually it can provide a lot of viscosity local to this region. So actually, in the protective effect around the villus tip might largely be provided by the DNA rather than the mucin. So that was quite a surprise to us. Now, what I've talked about has largely been done in vitro or in model systems, so certainly away from what happens in real life in a live animal. So we were interested to see whether you see the same sorts of behavior in animals. So we ran a pig study where we fed animals for 3 days either on a controlled diet or a controlled diet with 10% oat bran in it, so increasing the amount of soluble dietary fibre that these animals were consuming. The pigs were then slaughtered after the 3 days, and the intestines were collected, and the viscosity and permeability of the intestinal mucus was determined. And the first thing we looked at was the ability of 500 nanometer latex beads, as we've used previously, to diffuse in the two systems, and there was no difference at all in the mean square displacement, and thus not in the microviscosity. And we were a bit disappointed by that, but then I suppose we thought, well, actually that's quite a large size. Maybe we'd expect everything to be trapped. What about things that are a bit smaller and should in principle be on the edge of that 100 nanometer limit to be able to diffuse? So we looked at 100 nanometer latex beads, and well, I'm not sure whether it was to our surprise, but what we saw was that there were differences in the local viscosity, so the microviscosity, between the two systems. So the dietary fibre-fed pigs had intestinal mucus that was less permeable to 100 nanometer beads, and saw a significant increase in its viscosity. So clearly the dietary fibre there is important, at least for changing the rheological properties. Of course, we don't eat latex beads, we eat food. So we were also interested in seeing whether the increasing dietary fibre decreased intestinal mucus permeability to lipid digester. And we were pleased to see that it did. So the data showed that increasing the dietary fibre reduced the mass transfer of lipid through the intestinal mucus in these oat-fed pigs. So it looks like the things that we've seen in vitro are also seen in vivo. Of course, it's difficult to show directly, and this is work that we're looking to push forwards in future. So we've shown that in this case, increasing dietary fiber beta-glucan can alter the mass transfer of bile and lipids. So we know it can be used to alter cholesterol metabolism, so bile recycling. But this is the first time it's been shown for lipid more generally. The last thing that I wanted to cover was, so we talked about soluble fiber, was something on insoluble fiber. So in this case, looking at the fate of cellulose nanocrystals, it has become moderately popular to have a look at using cellulose nanocrystals, both alone and in conjunction with other components, as Pickering stabilizing agents in emulsion systems. So hence the picture here, you can see a lipid droplet covered in cellulose nanocrystals, so giving it this hairy look. And these are actually quite effective at stabilizing the emulsion. So once they are absorbed to the surface, they don't come off very readily. And so they're good at keeping the stability of the emulsion. Now, of course, under normal circumstances, nano and food are two words to be kept well apart, and people are a bit nervous of having nanoparticles in their food. Of course, in reality, everybody has nanoparticles in their food, and you can think of many systems that are on the nano scale that are very common to food systems. So, but nonetheless, people are nervous about that, and we wanted to look at what happens to the cellulose nanocrystals once you start to consume them. So initially, we looked at what happens through digestion. So in the gastric phase, you can see the emulsion droplets here in red, and you can see at least where it's thick, the layer of nanocrystals in blue under gastric conditions. And then if we go to intestinal conditions, where the lipid starts to really significantly be digested, you can see the lipid droplet here highlighted in red, and you can also see, I hope, the exciting range of self-assembled structures that you start to generate as a result of hydrolysis of the lipid. So from quite large structures close to the droplets to things that are clearly much smaller and actually most likely you can't really resolve, that are made up of fatty acids, monoglycerides, bile, and other components. So one of the things that you can see for sure is that the cellulose nanocrystals don't prevent the hydrolysis of the underlying lipid, and that also this kind of stream of lipid coming off the surface also removes a lot of this nanocrystals. So they don't hinder the lipolysis stage. What about what happens to them once they start to be absorbed? Well, here you see some tissue sections. The quality is not great because they've already been incubated in an Ussing chamber for us to quantify absorption. But nonetheless, I think what you can see is the villi here. We've stained the nuclei red, and again the mucus in green. So you can see it in the base of the between the villi here, but you can also see it across the tips. And you can see that on the kind of upper side of that, there's quite a bit of blue and essentially none down here. So most of the CNCs are entrapped in this mucus layer, which is unsurprising because they're relatively large to be able to get through that. So the main kind of take home really was that they don't pose a threat to the underlying mucosa, and you don't see sharp needles of cellulose sticking out of the cells in the underlying epithelium. Does it have an effect on absorption? Well, we looked at difference between saturated and unsaturated fat, and we found that in the case of saturated fats, there was a difference between the control emulsion and the CNC emulsion in the basolateral side. So we still need to do a bit more work in order to really understand what the mechanism of that is, but clearly there is an effect on the absorption of the saturated fatty acids from these systems. The unsaturated, we didn't see any differences. So it looks, in conclusion, like it's safe, and it may have some limiting effect on the rate of absorption of lipids. But as I said, we need to push that further. So, in conclusion, I think we can say mucus is complex. It's highly heterogeneous, especially in the small intestine, which makes it quite difficult to study and understand. Diffusion through it is governed not just by size, but also by charge. And also the heterogeneity can lead to significant penetration in addition to the particles being able to penetrate through that pore size and having the right charge. And we've also seen that flow patterns in the small intestine are the cause of that heterogeneity, and that they drive penetration between the villi, which helps maximize the diffusion and absorption. So we shouldn't think about the small intestine as having a protective layer of mucus over the villi in the same way that we have in the large intestine. And we've also shown that biopolymers, especially soluble dietary fiber, can diffuse into the mucus and decrease permeability, at least to certain things. And we've shown that insoluble dietary fiber will be trapped on the surface of the mucus layer and can also have some effects on permeability. So that was all I was going to say. So thank you very much for listening, and I'm happy to answer any questions.
Okay, thank you very much for that very interesting and stimulating talk, Alan. I hope there will be lots of questions I'm expecting from the audience.
I've got a couple to kick off with, if you like. One of the things that you're going to encounter in vivo, Alan, is the microflora. And I think all of your experiments are essentially tied up with tissue and so on, mucosa that has been extracted and looked at one way or another. But in vivo, you're going to have constant contact with the microflora, especially in the large intestine. Have you got any comments to tie up with that?
I think you're absolutely right, of course, and I think the differences in architecture are important for that. I think actually you say, especially in the large intestine, but I'm always quite surprised at the amount of free fatty acids that are generated in the small intestine as a result of microbial fermentation, especially more distally. And I think that that's where there is a careful balance in the body between permeability to bacteria versus permeability to other components, nutrients, etc., and mixing. Right. And why you see the secretion of antimicrobials more proximally in the small intestine, yep, because the mucus isn't providing that protective layer that it does further down, but yep, you can potentially have relatively high numbers of bacteria.
Okay. And a sort of follow-on question to that. Does it make any difference if you use animals to look at this? When we want to know a little more about the human situation. I know. I think pigs are probably the nearest to human, but they are different. And if you go,
yeah, no, I agree. They are quite different. I mean, one of the things that we were surprised at is the distribution of different types of bile acids. In pigs, very different to humans. Actually, very strangely, cows are much closer to humans in terms of their bile salt distribution. No, I agree absolutely. Start doing some of these studies.
There are lots of things that people do. We're not going to be sure that people for sure.
Yeah.
Thanks, Alan. Thank you.
No problem.
Thank you, Tony, for the question. I think we have Paolo. Paolo, do you have a question?
Yeah. Hi. Hello, Alan. Very great talk. Hi. When you talked about diffusion of these particles in mucus, what do you think? I mean, which is more supreme in controlling this diffusion? Is it the size or maybe the charge?
Okay, so obviously, in terms of the intrinsic requirements for diffusion, size is pretty critical. So smaller particles diffuse more quickly than larger particles, and anything over about 3 microns essentially doesn't diffuse at all. But interaction with the mucus as that protective polymer layer is also important. So anything that has positive charge will bind to that and not diffuse. It also has, as I'm sure Tony will support, hydrophobic patches in it, which means that hydrophobic components will tend to bind to it as well. So the ability of something to transport through that layer relies on diffusion of the object that's transporting through. And this is why I really talked about lipid, because lipid is intrinsically insoluble. So it only diffuses through as a self-assembled object. And that might be a small mixed micelle, but it might be a large multilamellar vesicle. And so the diffusion will be different for those systems. So that's why both of those factors are important.
Thank you.
Thank you, Paolo. I think we have Vincenzo next.
Alan was great, honestly. After the one last year, this was entirely different, but still very, 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 very good. So always, I learn a lot every time, and I have probably 2 quite silly comments. One is a statement that I say I really agree with your thought around the fact that saturated and unsaturated fatty acid might behave differently, and I think there isn't probably enough emphasis in the literature right now. I'm not very much on the digestion spectrum, but I work with fatty acid in different, so I think I absolutely agree with you. We know they have different emulsification capabilities, so you would expect some different behavior in the intestine. So this is one statement. For as much as it can matter to you, I completely agree on that point. So the second one, I'd say, probably you touched upon that, is the soluble fiber, you rightly say, they can penetrate into the mucus layer and slow down the diffusion. Do you have an idea of how long they can stick in there? Because I'm also almost thinking from the other side, if they close the pore for too long, maybe you can have some absorption impairment in some situation.
Yeah, okay, so that depends a little bit on where it is, but certainly in the small intestine, mucus turnover is pretty high. So this is in some ways why mucoadhesive forms of drug delivery are a bit limiting, because you stick to a mucus that's not going to be in place for very long, particularly in the small intestine. So even if you're able to penetrate, maybe that's not terribly relevant, because the mucus is coming out relatively quickly. Certainly when we talk about that mixed heterogeneous region, where you're seeing the villi come apart and then come together. So you're seeing material go in and out. Clearly what's coming out is pulling the mucus out as well. So you can understand why turnover of mucus in those regions is relatively high. I think it's less of a problem in the large intestine where you have these stratified layers. But then obviously you're having hydrolysis of fibre by bacteria. Actually, not just of fibre. There are lots of mucolytic bacteria in the large intestine as well, but they're mostly working on this loosely adherent layer, which is much more tractable.
Thank you very much, Vincenzo. Gleb, do you have a question?
Yes, I do. Hi, Alan. Hi, good to
Hi,
see
Gleb.
you, and fantastic talk. Just two very quick questions. So what I'm very intrigued by this microflows and the characterization. So as we know, the mucus starts as a vesicles, right, in the goblet cells, and then they get amalgamated. Do you know actually this evolution before it gets lost?
Sorry, Gleb, you'll have to repeat that. I completely didn't get any of that.
Gleb, if you can talk closer to the microphone, I think it is a problem.
I'm already. Can you hear me?
Yeah, I can hear you now.
Yeah, just switch that tornado off.
All right. Oh, no, I've muted Tony. So Alan, a brilliant talk, and so the question I have is that the mucus starts as a vesicle, right? Goblet cell. So my question about the do we know the degree of amalgamation of the vesicles when they kind of get fused together? So do we know how much of the interpenetration of the material between the original vesicle goes on by the end of the sort of mucus life as it gets sloughed off? Yeah,
in principle we do. So there have been some models from the kind of Johansson group that have looked at that. So the key is that in that kind of vesicle, as you called it, you have a tightly folded package of mucin that rapidly expands significantly. And they claim that it's folded in such a way that when it opens up, you maintain that hexagonal structure, right. And that builds up the kind of layered structure, and that there are other binding agents essentially that link those stratified layers together. Right. I think the evidence for that looks quite good in the large intestine. I think in a small intestine, much less so. If you try and look for a stratified layer, there's a little bit of evidence really close to the villi, but it's poor, I would say, generally, because of the amount of mixing that goes on. You don't get the formation of the same kind of structures that you do elsewhere.
Right. And another quick question I had is just building on the work we were done with in Queensland with Jason about the cellulose nanocrystals and how sensitive they are to the salt environment. So first of all, have you observed the effect of the salt on the digestibility of the emulsions? And then is there any strategies that you apply for, let's say, crystallizing these cellulose nanocrystals at the interface in a different way? So I know that they form a liquid crystals and they can form a 2D crystal as well. And there are some reports that report the emulsion that are relatively random, and there's that are more ordered. So can you elaborate a little bit more on that?
Yeah, it partly depends on how you form the crystal. So the degree of sulfation is critical to their ability to be to have any surface activity, really. And so we work with the guys at Nantes in Nantes, who produced the nanocrystals, and they had some crystals that were pretty surface, well, relatively surface active. At least once you got them on the surface, they would stay there. Whereas colleagues in Leeds, Brent and Anvesha, have been working with similar system, but theirs wasn't quite as good. So they found they needed to include some protein in there as well to get a really stable emulsion system.
Right. Okay.
So it does make a difference. In terms of the salt on digestion, we've tended to use just physiological conditions. So the kind of salt conditions that you would expect to find under normal circumstances in the GI tract. I mean, I guess you can increase the ionic strength or decrease it. That's bound to have an effect, obviously, because you're reliant on charge to a certain extent to build the density of that network.
Absolutely. It's kind of like one of those things that, like, especially with the foods that, like with the vegetarian or vegan foods, sometimes they add a lot of salt, you know, just to make it taste better. And you kind of think how that would affect just even when it travels as a bolus, if there is some, of course it is a small molecule, it is ions, they diffuse pretty fast to equilibrate, but still kind of it could be that it's a pretty sort of high sort of concentration of different.
Yeah. I mean, it depends a bit on I mean, we've been looking a little bit at the bolus formation that you have, not just in the mouth, but in the stomach, and how it's persistent. And colleagues in the Netherlands have been looking at the ability of acid to diffuse into protein gel systems. You'd think protons would diffuse pretty quickly. But because of the high buffering capacity, actually the diffusion is quite slow. So I can imagine that if you have any interactions in these systems, then ionic diffusion is also going to be pretty slow.
Thank you. Thank you. That's brilliant. Thank you.
I think, David, you have a question?
Yeah, thanks, Vlad. Alan, thanks very much for the really interesting talk. I guess being a Scot, I love my porridge. I'm just interested in, you know, what nature was the oat bran given to the pigs? Was it just basically, you know, rolled oats? Was it cooked in another way, or?
It wasn't cooked in another way. It was standard oat bran, as you might buy yourself. I would have to look back at the details of precisely what it was we fed, because it was a little while ago now. But it had a reasonably high concentration of beta-glucan in it.
I was just wondering if, you know, I think I don't know if there's a school of thought that believes that, you know, when you take oat flakes and you actually, you know, hydrate them and cook them, you get more beta-glucan out, you know, than you would normally, because it's stuck in this interesting cell wall material. So would you anticipate that you might get more effect and more bang for your buck if you actually, you know, make porridge and feed them porridge first?
Oh yeah, I'm sure you would. Yeah. No, I with my Scottish heritage, I'm a big fan of porridge as well. I think that has lots of benefits.
Yeah. No, thanks. I'm just curious about that other processing step that might have.
Yeah. No, I completely agree. I think the processing would be beneficial.
Yeah. Well, thanks, Alan.
Thanks, David. Can I just ask a quick question? It's not really a question. Sorry, Steve. It's so you have all the soluble and non-soluble dietary fiber, things like cellulose nanocrystals, which disrupt the things like permeability. And as they move further down the intestine, where there's a lot more bacteria, will that also have an impact on the bacteria kind of interactions and production of metabolites that are useful for things like the gut-brain axis? And could that have an effect on nutrient absorption through the disruption of the bacterial metabolites?
The short answer to that is yes. In my experience, nearly everything affects the colonic bacteria, assuming material gets that far. So certainly changes in diet and dietary fiber will change that. Now, whether you change things positively or negatively depends a bit on the precise nature, but I think in general, we don't consume enough dietary fiber of any form in this country. So I would say anything that improves the amount of dietary fiber that we're consuming and the amount that's reaching the colon would be good. And that's likely to be beneficial in terms of the spread of bacteria that you had that are also able to metabolize other things. So particularly a lot of the kind of phenolic compounds that we consume, the bioactive form is only generated by gut microbiota. So for sure, yeah, it's important.
Thank you, Alan. Steve, do you have a question?
Alan, yeah, so I've got to go in a few minutes because I've got another meeting at 1, so...
Me too.
It's Gleb's tornado again, I think. Right, I don't know whose tornado that was. I think it's Gleb. But yeah, thanks.
Steve,
Someone got the mic on. I don't know. Some interference coming from somewhere. But anyway, just to say thanks ever so much, Alan. That was fab. And I just thought of a quote that Roger Payne once said. It was about mucus. He said that if it's love that makes the world go round, then it's mucus and slime which keeps it in perpetual motion. And that certainly applies to the gut. But before I go, I mean, it was a wonderful tour also of different labels, all these pretty labels of mucus and components. And I think my favourite was the Oregon green,
Yeah,
Oregon green one. But when you're doing the FRAP measurements, do you have to label the alginate and things there with some sort of fluorescence label? How do you? What do you use for that? Fluorescein or?
we usually don't.
Oregon green.
Yeah, we depends. We usually don't use fluorescein directly. We have used it for FRAP. It can be quite good for that because it bleaches pretty readily. But for many of the other things, it bleaches a bit too readily. So we have used Oregon green. For lipid, we typically use Nile red. And for the CNCs, I think we use Calcafluor, which is a cellulose label. So there, I mean, the range of dyes available these days is quite amazing. In fact, now you have these asymmetric dyes that are able to measure local viscosity. So the range of stuff that's available is quite amazing.
Great. So Oregon green is fluorescent, so I'll make a note of that.
Oh yeah, yeah. No,
I like the blue one as well. The blue one looked quite nice. The Pro, whatever it was called.
Oh yeah, yeah.
In fact, we need to have a word with you. I'm going to go shoot a minute, but about we've started these lipase beta-glucan interaction experiments again. So I don't know whether Neil's still with you, but maybe later I'll give you a call.
Yeah.
So yeah, thanks. That was great. Thank you.
Thanks very much,
Steve. Thanks, Vlad. I thoroughly enjoyed giving it. Thanks for all the questions and thanks for organising it. It's been great.
Thank you.
Thank you.
Have a great day, everybody.
Okay.
Thanks a lot.
Thanks, Vlad and Steve.
Yeah, thanks everyone.
Yeah, thanks. So I've got a dash now. I've got another one.
Bye. Bye.
We'll do our class at 2 o'clock. See you.
Okay, see you guys. Tony, I guess we didn't have much time to talk about some of your flyer.
Yeah, that can go over to next week. It's literally this week that we started on that. But if everyone has the flyer, then yeah, I'll come in next week and make a comment on that.
Yeah, that's brilliant. I think next week we don't do. Let me just check if we have any speaker. I'm not sure if we do, which means we'll probably have. Oh yeah, we have Gleb actually. Yeah, so we have Gleb. Hopefully, it won't take as long as today's, but hopefully we'll have some time to talk about it next week.
No, that's good.
Definitely.
Thanks, Dan.
Brilliant.
That's great. We'll sort that out next week.
Okay, fantastic. Thank you very much, Tony, and have a good week.
Yeah, I've got to leap off now as well. So
cheers.
See you next week.
See you.
Bye-bye.
Bye.
Bye.